Welcome to the Jones Seminar. We are featuring a speaker today from UC Merced. It's Professor Roberto Andresen. He, and as you should know, Professor Alex Boyes and I all went to Cornell together. So this is a little bit of a fun reunion for us. And really excited to hear about his research on adhesion. So, like I said, Roberto completed his PhD at Cornell in material science and engineering, did a postdoc at Urbana-Champaign in Santa Barbara, where he worked with, I'm going to screw up the last name, but Jacob Iacovelli, who is like the founding father of this type of adhesion science. So really, really cool legacy there. And Roberto's taking that to the next level. So really excited to hear your talk today, and thank you for coming.
Thank you, Emily, for the invitation, Alex. Thank you very much for the invitation. And thank you all. Thank you for the welcome. Don't forget to sign in. Good. It's been a minute since I think on this part of the country since I left Ithaca. So thank you again for the reunion. So we reconnected actually through meetings, the Adhesion Society meeting last year, the Biomedical Engineering Society last meeting. So definitely worth attending meetings at any stage in your academic career. Not only for science, but also interaction, social interaction networks. So I'm in chemical and materials engineering at the University of California, Campus Merced. I'm going to spend a minute introducing you the institution. But first, I want to start with acknowledgements. This is the group. The work that I'm going to be presenting, I want to split it in two main topics, each of them with two short stories. One on adhesion, and collaborating with Alison Butler from UC Santa Barbara, with Ying Yan from Yale University, and Andrea Merced from UC Merced. And then the second part, if time permits, I'm going to talk about not necessarily, or not strictly speaking, joint tribology, but so our initial efforts to model the articular cartilage surface or the cartilage interface with synovial fluid, and that's in collaboration with Kevin DeFrance at Queen's University. So this is the group currently. The work I'm going to be presenting has been conducted mostly by Tajin, but Ume, Diego, and Sishin. And of course, I want to thank funding sources, LSF Career for the joint tribology work, and DARPA for all the adhesion work, together with support from the institution UC Merced and the NSF CREST Center, CCBM, that has also supported us initially. So these are a few images from the region. So UC Merced, this is an aerial view from spring. So everything is green. In summer, everything turns yellow, brown, extremely hot. And campus has been growing. We reached R01 status a month and a half ago, so that was a big accomplishment. And this is where our creative spaces are located, our laboratory spaces. I enjoy spending a lot of time here in the mountains, the High Sierras. We have peaks of 12,000 or so, no signal, really nice place to forget about emails. But you have also, of course, a lot of really nice outdoors here. So going to what we are interested, at some point in my academic training, I came to, someone introduced me to the concept of interfaces. And it was like, this sounds actually interesting. And I know many of you work with interfaces. So this is an interface from one of my hikes, Skeleton Lakes at Yosemite. So we have, of course, a solid phase, a liquid phase, and we have vapor all over the place. And as many of you do, what we try in an experimental setting or also in computational settings is to reduce these complex interfaces to simple, well-behaved and controlled systems. So we have, of course, a solid, a liquid, and a vapor-air interface here. So we're trying to do something similar with biological-inspired interfaces to understand how molecular rules are mediating, on one end, adhesion, and how molecular rules are, as well, regulating tribological performance, particularly synovial joints. Okay? So I'm showing you here two schematics. Now, this is summarizing really the two topics that I'm going to talk about, the two stories. Biofilms and how particularly molecules secreted by bacteria or by biofilms can be used to understand molecular rules that allow to develop technologies for wet adhesives. And on the other hand, I'm going to introduce you some concepts and some molecular players that are important in understanding the tremendously robust tribological performance of synovial joints. We have 600 or so synovial joints. We never think about them unless we start to age and get osteoarthritis or rheumatoid arthritis. Good. So I'll start with biofilm as a source to understand wet adhesion. And some of the challenges that we can find in wet adhesion is, of course, that compared to vacuum conditions, van der Waals forces in aqueous environments are reduced. We have also ions that are going to screen electrostatic interactions. We have hydration layers that will inhibit intimate contact of the moieties with our substrates. And depending on the environmental conditions, we may have hydrogen bond donors that oxidize. Okay, so we lose the contributions of all these non-specific interactions, weak interactions. Good. And a little bit of history. And many of you have heard or know about DOPA. And this molecule was first reported in '81 by Herbert Wade. And that triggered a wave of research around this moiety, DOPA. And DOPA, which stands for dihydroxyphenylalanine, it's a post-translational modification of tyrosine, including an additional hydroxy group to the ring. DOPA became, of course, of extreme interest to many polymer scientists because of the wide range of interactions. Of course, we have, in the context of marine muscle food proteins, which I'm going to elaborate in a second, we have interactions that mediate organism-substrate interaction, extremely robust adhesion, and these molecules can also mediate cohesive interactions, okay? We have coordinations, we have cationic solid interactions, we have covalent bonds, we have, of course, lysine, DOPA synergies to increase adhesion, we have hydrogen bond donors, which will contribute to adhesion, and we have pi-pi and pi-cation interactions. So I'm going to focus on the context of adhesive. Okay. So marine mussels have been used for decades since the report by Herbert Wade as the prototypical example of one of the prototypical marine organisms to understand wet adhesion and fight all these challenges that we can find in aqueous environments. You can think that the dielectric constant, for example, will be one of the multiple parameters decreasing the interaction potentials. Okay. In 2015, and I'm showing you now this molecule, which has something similar to DOPA, okay? In 2015, Mayer with Alison Butler, with Jacob Zefalashvili, they synthesized this molecule. It's inspired in cytophores. Cytophores are also bacteria-secreted molecules used to collect iron and transport iron through the membrane of the bacterial membrane. And of course, they identified that this catecholic moiety is very similar, not the same, but very similar to the catecholic moieties present in marine muscle food proteins. So we have here the ring with the two hydroxy groups. We have here a lysine, and we have here a core. And three arms, very simple molecule, well behaved. And I'm not going to elaborate, but this simple geometry reduces the complexity of conformational, the conformational complexity imposed by marine muscle food proteins, which are much larger molecules. So in that paper, they showed that this catecholic moiety performs as a robust adhesive only in the presence of an amine, the lysine molecule, okay, or moiety. And if you remove one of the hydroxy groups or remove completely the amine from the molecule, the addition, which is summarized here, the addition energy for the different molecules, decreases. So very sticky, these two red bars here. No catechol or no amine, no addition. That's the summary, okay? So they used an apparatus to measure all this interaction or this pool of forces or pool of energies. They used a device really pushed and developed by Jacob Relashvili, Jacob Klein at the Weizmann Institute as well. They call them over there the Surface Forces Balance. We call it here the Surface Forces Apparatus. It's a box that has this geometry. It has these components. It has a drive here. We have a double cantilever spring which buckles rather than bending. That allows perfectly vertical displacement. We have two macroscopic surfaces that are 1 centimeter in diameter. The surfaces are semi-cylindrical surfaces, like my fingers. They're mounted in a cross configuration that resembles a spherical contact against a flat. They're mechanically stable. And one of the surfaces is mounted on this double cantilever spring. The other surface is mounted on the holder, on a piezo, on the actuator. And these two surfaces, I'm gonna move to this slide, build an optical cavity. And I'm gonna explain the optical cavity in a second. With this system, we have force sensitivity between these macroscopic surfaces of around 1 nanonewton. We have a distance resolution between the mirrors of 0.1 nanometers, and we have motor displacements from 0.5 to 100 nanometers per second. So we have a wide range of velocities with extremely high force and distance resolution. And this distance, I have to point, is absolute, as opposed to other force spectroscopy measurements like AFM. So the optical cavity, so this is how we measure the force. I'm gonna have one more plot explaining. So we have the double cantilever, it buckles, we measure force. This optical cavity allows us to measure the absolute distance between this mirror and this mirror with this resolution. So we have mica, thin sheet of mica, and aluminosilicate. It's atomically flat, and it's chemically inert, in the conditions that we use them. So we have silver deposited on this mica, and then we have a medium. We place these two surfaces containing the mirrors, and we shine white light here through these surfaces. We have a constructive interference, and only those wavelengths that yield a constructive interference will exit the cavity once they reach an escape intensity, and we direct these wavelengths to a spectral analysis set of components. We have a spectrometer, some cameras, and then we obtain this type of interference patterns. And we use them to measure the distance, as I was mentioning. So we have here the interference patterns. We have the geometry here, the two surfaces at a finite separation, this gap here. We have the silver, the mica. We have our aqueous medium. It can be vapor as well, of course. We can have gels. And these interference patterns will map the geometry of our surfaces. When we bring the surfaces into contact, of course, these are elastic solids. They're gonna elastically deform as long as we don't press too hard. And we can map the geometry of that interface in real time through these interference patterns. We can also measure the displacement of the interference pattern. So we have the wavelength space here. We monitor at any given instant the position of these fringes. And we relate the position of those fringes to a reference state. And with that, we can obtain the absolute distance, meaning that we can measure the absolute thickness of the medium that we're confining. We can also extract optical properties, like the refractive index of the medium, how the refractive index of the medium is changing as we confine. Okay, I'm not going to talk about that. We're going to focus on distance. Finally, to summarize, so we have the drive, the double cantilever spring represented here by this extension compression spring, the surfaces at large distances, no interaction potential, or the interaction force field is weak. We cannot really measure it. And as we come closer, the interaction potential is large enough that we can measure it. And in this case, we have a jump in, and represented in the negative quadrant, attractive forces. We can, of course, compress. We are now in a repulsive regime. We separate the surfaces. We reverse the motion. There's going to be a jump out. I'm going to be reporting mostly or talking about the pool of forces, depending, this can be an adhesive force, it can be a cohesive force. And with that, we have, of course, we reconstruct interaction potentials, and we can, using the jargon approximations, estimate the interaction energy and compare it to that of flat plates. Well, because geometry, of course, in all these contacts matters. So building on that molecule that I showed you, Mayer reported in 2015, so I started working with this molecule together with George Deegan. So we have, again, the same molecule. It's a little confusing. I'm going to walk you through it. So we have the Trent scaffold, aramine core, and this is the geometry and the chemical moieties that were reported previously. We have the lysine, this cationic moiety, and then we have the catechol. Remember, these locations are not the same as in marine muscle food proteins, yet sticky. Now, the question was, does increasing the distance between these two moieties abolish addition? And to do that, so we designed this new architecture, this new molecule, in which we have the catecholic moiety, we have the lysine, and we included a few glycine spacers, okay? And we know in proteins, peptides, these glycines will provide certain conformational flexibility. But what is important is that with these glycine spacers, we could swap the position of the amine, of the lysine here, without changing the molecular weight of the molecule. So here's another example. So we have these glycine spacers, and in summary, so we did, of course, a good number of measurements, all using the Surface Forces Apparatus, and I'm showing you the summary plots. So we found that increasing, this is the original moiety, we have the high, extremely high pool of forces, now reported as the negative of the pool of force. And as we increase the distance between the catechol moiety and the lysine, the addition did not disappear. And we designed molecules up to 6 glycines in between, but we never saw a loss or abolish addition, just a decrease in the addition. The way we are interpreting this is that increasing the molecular size is reducing the number of molecules participating in the bridging event of the two surfaces, and therefore this reduction. Now, the interesting part of this is that if you have molecules with only the with only the catecholic moiety, and in the same solution, the molecule with only the lysine, the cationic moiety, there's no addition, there's no synergy. So both moieties have to be present in the same molecule in order to be sticky, okay? So this is the first part. Now, something I haven't told you yet is that these molecules, of course, have these catecholic moieties. Catechols have been used very successfully in patented adhesives for concrete, fillers, dental adhesives, but these catechols are extremely sensitive to pH changes. So they perform very well as sticky molecules as long as the pH is below 5.5. So they like acidic conditions. If we go past 5.5 pH, they start to oxidize. They very easily auto-oxidize. So that limits the application of these molecules. And we can see it here. pH acidic, or pH 3.3, extremely sticky, and now as a function of contact time, contact mechanics, of course, play a role here. We're increasing the number of bridges as a function of time. And we increase the pH and we see a dramatic reduction in the stickiness. So we designed a couple of additional molecules, and I'm going to show you two. One, just for completeness. No, I think we anticipated what we're going to observe. We synthesized, together with Alison Butler, a new molecule. They synthesized a new molecule. We characterized the molecule. But now with the hydroxy groups located in the same positions as we find in the rings in marine muscle food proteins. So this is one new molecule. And second molecule, Marcus Textor at the ETH several years ago demonstrated that if you incorporate an electron withdrawing element to the molecules, you can extend the range of pH values over these molecules remain stable and mediate stickiness. Okay, so let's do that. So let's change the chemistry of this moiety here and add an electron withdrawing element, okay? And then we have this HOPO molecule. And this was also inspired by Craig Hawker from UC Santa Barbara, who reported stability of this moiety in the context of hydrogel chelating iron. Good. So let's have these two molecules. We're going to test them. And we're going to, of course, explore how they perform as an adhesive molecule under different pH values. The first thing is we prime our surface, or we inject the buffer, clean. We inject our molecules of interest. We tested pH 3, pH 7, pH 10, 150 millimolar, physiologically relevant conditions, and when we performed our addition measurements. So the first thing was to optimize the concentration for maximum addition. So with the SFA, since we can measure the absolute distances, we can get a relatively good idea of what regimes of concentrations, based on the film thicknesses, are leading to an adhesive interaction, or failure, I have to say, and which conditions, of course, correlating forces with the film thicknesses, correspond to cohesive forces, okay? Of course, we can do a lot of surface analysis to validate this, but based on this, we believe that the adhesive regime is in this region, and as the film thickness increases, meaning that more molecules are building a multi-layer film, this addition decreases. So these molecules we interpreted as they're not good cohesive molecules, or they're not molecules mediating strong cohesion, but they're good for mediating addition, okay? And we are thinking this as having two half films on each surface, and when we bring the surfaces, we are maximizing the number of bridging molecules. Good. So a couple of force distance profiles. So I showed you already an hypothetical force distance profile, and these are now real force distance profiles. We have the distance, and then we have on the y-axis the force normalized by the radius of curvature. We always normalize by the radius of curvature, again, because geometry matters. And we measure also in real time for experiments, since we have the interference patterns, we can measure the real radius of curvature for every experiment and at every given instant. So we have first high pool of forces, or no interaction, we compress, and then we separate, and there's a jump out here. For pH condition pH 3, at pH 7, the addition decreases, and at pH 10, the addition decreases. And this is, I'm only showing you one of the three arms of the HOPO moiety. So that moiety, that is, in principle, capable of resisting oxidation at wider pH ranges. So summarizing a good number of experiments, and also as a function of time, and adding a reverse step here. So we have the pH 3 adhesive performance, really good, not as good as the catechol moiety. Remember, we were at these values, but still very robust addition. We increase the pH value to 7, and we retain a good fraction of that initial addition. We went then to pH 10, so this is fully deprotonated molecule, and we see a dramatic drop. But what is really interesting and exciting to us is that after switching the pH back to 3, we recovered a good amount, a good fraction, about 85% of the initially measured addition. Some of you may ask, like, can you repeat this over and over? We tried it, but these systems can get very easily contaminated. Look at the distances that we're measuring. And I pushed my postdoc to do it several times, but there's a limit to it. Okay. Second moiety, this is not going to be surprising to any of you. I mean, we have the new molecule that has this now muscle-inspired catechol rather than the styrofoam-inspired catechol. and we repeated the same experiments at pH 3, not surprisingly extremely sticky. But as we change the pH to higher values, it oxidizes and we lose all stickiness, okay, or addition. And similar plots as a function of the contact time. At pH 3, extremely sticky. And at pH 7, it oxidizes. pH 10, really no dramatic change. And after switching back to pH 3, we recover a fraction, we think, due to the recovery of the positive charges. Mica is negatively charged. So what did we learn from these studies? So we have that the cyanophore-inspired molecules, DHP and HOPO, adhere strongly to mica in acidic conditions only. HOPO showed robust adhesion at pH 3 and 7, so expanding, for example, for biomedical applications, potentially. And recovered adhesion after exposure to pH 10. And what we are working on right now is, can we identify or the synergies that have been already identified with catechols, for example, lysine, if we incorporate other hydrophobic moieties like phenylalanine, the addition increases dramatically, exponentially. Are those synergies that have been identified also applicable to the HOPO moieties that are, we know now, more robust against oxidation? So that's what we're working on right now. So this is the first part on addition. So moving on, the second short story on addition. Any questions so far? Good. So moving to the second part, still addition, stickiness. So I'm not going to call it addition, or if I call it addition, I don't really mean addition. It can be cohesion, addition, or a mixture of that. So I'm going to try to call it stickiness, okay? Because we don't know yet if it's adhesive or cohesive or a combination. So this is working with Yin-Yang and Andresen. And in 2019, Rich Olson, and then later on, Yin-Yang and Rich Olson confirmed that a very strange and unique behavior of Vibrio cholera around this protein. So I'm showing you here BAB1, it's a bacteria adhesin of Vibrio cholera, and this adhesin is required for Vibrio cholera to form a biofilm and colonize the gut, okay? And what they showed is, in these two papers, is that if this region of the protein, we have the beta propeller domain reaching in beta sheets, we have this beta prism, and there's this loop here. We have this hairpin that if it's deleted from bacteria, the bacteria biofilms cannot form, okay? And in solution, if you remove the domain here, the protein becomes water soluble. With this moiety present, the protein is extremely difficult to solubilize in aqueous solutions. So good. We started working with Yin-Yang, and let's look at this moiety as a potential new template of sequences that can mediate stickiness. So the sequence of this peptide here is shown here. I'm summarizing some of the hydrophilic domains, but we have three constructs mostly. We have this 57 amino acid wild type construct. We have a central portion peptide, much shorter, extremely rich in hydrophobic moieties. And then we have this short domain that has, interestingly, also hydrophobic moieties and also some lysines present. And similar to what we did with the HOPO with the smaller and cyanophore-inspired molecules, we injected our peptides of interest. It can be the short, the central portion, the full length. And we worked at a buffer of 190 millimolar in pH 7 or milk water. I'm going to explain a little bit about that. And we're using this buffer because that's the buffer that is commonly used in biofilm research. And then we performed our addition measurements. And this is the geometry, and it's important because we're working with a symmetric configuration, meaning we are decorating both surfaces at the same time. We can change that configuration and do an asymmetric and only deposit, but that is for another day. So now I'm going to jump straight to the summary plots. I've shown you already a lot of force distance profiles, so there's no need to repeat that, but we perform the same. No? We are measuring the distance and we are calculating the force as a function of that distance. So we have now, we're calling it dwell time instead of contact time, but it's the same. and then we have the pool of force and the pool of energy here. So for the wild type, the full sequence, we see that the moiety is very sticky. Comparing to the previous molecule, we were here. But we call very sticky anything that is above 5, okay? So it's extremely sticky. And of course, we did a bunch of measurements with a short peptide. We have a wider library. And the only peptides from these libraries, and I'm explaining or pointing particularly the short and central portion, only the central portion was sticky. And it was surprisingly as sticky as the wild type peptide. And they behave very similarly. This huge error bar tells me that they behave very similar as a function of contact time. So that is interesting. So we believe that we have identified a region, there's some hidden synergies that we need to narrow down to fewer amino acids and identify or see if they are as sticky as the short or the full-length sequence. Now, this plot is a summary of addition or stickiness measurements of only the central portion of this larger sequence. And we see, and they're now for M9, so high ionic strength or ultra pure water. And the performance of these two conditions is very similar. We have not been able to replicate measurements from this wild type in water conditions. It has been extremely difficult to work with aggregates. This extremely hydrophobic rich content molecule is hard to solubilize without significant content of DMSO. And of course, we don't want to have this organic content in our eco solution. So we are doing a bunch of parallel studies to identify those conditions in aqueous solvents that will allow us to continue working with this wild type. But given we have identified, we believe, the responsible domain, and we can work with it in aqueous and solubilize it, I think it's already a win for us. Good. So summarizing, closing this second stickiness or addition, wet addition part. So I have here some information about the Vibrio cholera-inspired adhesive. We have BAP peptide, which is approximately 7 kilodaltons. And on the right-hand side, we have one peptide from marine muscle food proteins, which at some point I mentioned has been one of the prototypical peptides from marine organisms. Here's the sequence, MFP5 particularly, which is from the 20 plus MFPs that have been reported. This is the one that has the highest DOPA content, okay? The stickiest one. So we have a good amount of tyrosine. We have a good amount of tryptophan and phenylalanine. So very rich in hydrophobic moieties. And we have lysine, this cationic moiety that I've discussed in the past. There's synergy, okay? Lysine has to be present. And if we have some ratios between these hydrophobic moieties and the charge cationic moiety, is similar to the DOPA-lysine ratio. It is important to note that this DOPA, and I mentioned it already, is a non-canonical amino acid, meaning there's a post-translational modification or an enzyme involved in transforming this amino acid, canonical amino acid, to incorporate an additional hydroxy group. One of the advantages here is that we are dealing with only canonical amino acids, meaning that there's no additional work to do beyond the chemical synthesis, much simpler to obtain. So in this symmetric configuration, and we're working with we have results on asymmetric configurations, lower concentrations, and now comparing or trying to compare apples to apples. SFA measurements, mica, and similar conditions. High ionic strength, pH of 7, which is more relevant for biomedical applications, for example, pH of 2.6. The peak pool of forces, 20 millinewtons compared to the 7 millinewtons, okay? So I think we have a good template, and of course we are continuing to explore this. This is work, we're preparing the manuscript on this. So summarizing, BAP1 peptide pool of forces is higher than the reported one for MFP5s. So we have more than 40 years working with these catechols, and it hasn't really given all those promises. We have BAP1 biochemistry stable at neutral pH, also only canonical amino acids, And we have not identified those conditions that can allow us to differentiate between adhesion and cohesion. Again, working on that, but these aggregates are not helping. And the idea is to identify new chemistries. Good. So that is the first part on stickiness. So I'm going to stop here for a minute and ask if there are any questions or clarifications before I move to the second theme that's going on in the lab around synovial joint mechanics. Yes?
When you're talking about the synthetic engineering to get to work, how long does that stay sticky for? Have you done any degradation studies before?
That is a good question. We have not done any degradation studies. Typically, these experiments take 48 hours to complete. So within those 48 hours, good, as long as we don't have contamination. So we're always fighting small aggregates, particulates, and we can see that immediately in the interaction potentials. So we need to discard it. Particularly for the first part, we have to remove the surfaces to replace the buffer. So that increases, of course, the risk. But so far, around 48 hours.
So you said it's a symmetric test. So you're filling, correct me if I'm wrong, but you have both sides that contain this molecule, correct?
Yes?
And it's like the adhesions to itself, I guess, the cohesion.
Yes.
Have you looked at it just like in an asymmetric property where you have it maybe interacting?
Yes, and again, we don't know. Here it can be cohesion, adhesion, mixed, probably. We have looked, we have a set of measurements, but we have not been able to reproduce it enough times to be comfortable to report it yet. But we have one surface that is bare mica, and then we deposit a film, and we see a slight increase in stickiness compared to the symmetric configuration. Yeah?
Really awesome molecules right there.
Thank you.
So I was going to ask was like, have you tested any gallic acid components? Because gallic acid has 3 hydroxyl groups instead of 2 in dopamine. And so I researched a little bit about it, but any insights on that?
We have not tried it. That's a very good point. No, I mean, it's worth definitely trying it.
Okay.
Adding these moieties, no? Yeah. So let's find, again, no, we don't synthesize. If you're interested in synthesizing these molecules, I would be excited to work with you. Yeah. Other questions? Good. So now I'm going to move to synovial joints. So this is the second theme. I've been interested on this system for several years now, and really at the molecular scale. So what we are interested in is in modeling the single asperity mechanics of synovial joints. Of course, cartilage is extremely rough, it's porous, the joint is complex. So we have bones, we have the patella, and these bones are cut by a soft tissue, which we all know, it's called cartilage, and cartilage is more complex than we actually think. So there's like everything in nature, there's a gradient, and gradients matter. So we have a gradient in mechanical and chemical properties. Bone, calcified cartilage, tide mark, also another interface, radial zone with the collagen type 2 are aligned in a particular orientation perpendicular to the bone. And then we have a transitional zone, and then we have the superficial zone, and on the top we have synovial fluid. And I'm mentioning cartilage 2, collagen type 2, and that's the important isoform. In humans, we have 28 different isoforms, and they all have very different affinities for other biomolecules, and that's important. And here at the top, of course, we have a very specific architecture, orientation of the fibers. We have something green that is fibronectin. I'm going to speak a little bit about fibronectin. And then we have synovial fluid. And we are interested in that interface. So what's happening at the synovial fluid cartilage interface? And because depending on the lubrication mode that we're talking, I'm going to focus on boundary lubrication. That is when the two bodies are in intimate contact and sliding against each other. Well, it's practically a journal bearing, and depending, of course, on the viscosity of the fluid, and you can already think that in pathophysiology, the viscosity of the synovial fluid will change, and that will change the tribological performance. So this is taken from Tribology International, from Maxic, And this is for a classic metal junctions, but similar phenomena can be seen in soft cartilage materials, no? Where we have different surface potentials depending on the composition of the cartilage surfaces. We have, of course, third body. We can have cell debris or aggregates of proteins. We have cracking and fretting if the mechanical forces of the tissue can tear. The wettability or the interactions of the molecules are going to be different. So very analogous phenomena. And what happens, without going into much detail, is that when the surface properties, chemomechanical properties of that interface start to change, the friction coefficients will start to increase, leading to strains that deep embedded cells that maintain and repair the cartilage, called chondrocytes, are going to sense, reprogramming the cellular behavior of the cell. So genes will be activated and they will start to secrete components, extracellular matrix components, that should not be secreted and deposited to the cartilage tissue. So rather than collagen 2, which I said is the relevant component, will be collagen 1, more glycosaminoglycans, dysregulation on glycoproteins. And eventually, these are images of tissues, a healthy cartilage section, and a damaged osteoarthritic cartilage section. So you can clearly see how the cartilage gets damaged, and the chondrocytes at some point will not be able to catch up and keep repairing the cartilage. Of course, this is a very simple description of what happens in pathophysiology. I want to introduce two important molecules. So that's for the first study. So we have glycoproteins on one end. Particularly, we are interested in lubricin, which is a bottle brush. We have heavily glycosylated mucinous domain with specific binding domains to cartilage or for self-aggregation. So this has been identified as the main boundary lubricant. And we have glycosaminoglycans, non-sulfated and sulfated. They form these large complexes that at the end resemble also a much larger bottle brush structure. And I mean, there's a fun story here between Swan and McCutcheon. McCutcheon in the 70s reports that it's hyaluronic acid, that component responsible for the wonderful tribological performance of synovial joints. Later, Swan says, like, McCutcheon, you're wrong, and they start this scientific dialogue, each fighting to see which of these molecules is actually the responsible for this tribological performance. At the end, what we know today is that they're both equally important. It's the synergy, not only between these two molecules, it's the synergy with many other macromolecular constituents that give rise to the fabulous tribological performance of our joints. So it's a complex system. Between lipidomics, proteomics, we have more than 2,000 components in synovial fluid. So you know the SFA. This is yet another representation of the SFA. So we have our mica surfaces, mica, we have our spring with the motor, and we do this. But now, rather than stickiness or adhesion, so you can see that there's nothing in this quadrant, I'm going to talk about only repulsive interactions. And that's what you want in a lubricant. You don't want these lubricating molecules to breach the surfaces. That will increase the lateral forces to allow relative motion. So only positive, repulsive forces. So we inject synovial fluid into these mica surfaces as an extremely simple model of the articular cartilage. And we can discuss if it's a good or not representation, but we are interested in the synovial fluid mechanics. Okay? Mica negatively charged, mica and cartilage are both negatively charged with similar charge densities, so we can get away with that. So there's a good amount of macromolecular constituent absorption to the mica, dense, and then we have a slightly more dilute synovial fluid at the center. So we compress, you know these interaction potentials, how they look, large distances, and now I'm plotting this in a semi-log plot. And the interaction potentials start around 200 nanometers. And I'm showing you here three conditions. So a non-treated synovial fluid, so that is synovial fluid as extracted. This is equine synovial fluid from horse. Of course, we clean it to remove cell debris, and we see a particular profile, so here in red. in solid circles and out open symbols. And of course, these two curves overlap. And simple interpretation, there's no hysteresis. Now, the two other conditions, we used enzymes to cleave different, selectively, synovial fluid components. In the black one, we use the hyaluronidase to cleave glycosaminoglycans. The fragments are still there, so we're just reducing the molecular weight. We are not purifying and removing the fragments. But now the interesting thing is that we compress. This is the compression or the loading cycle, unloading cycle. And again, very simple interpretation, there's huge hysteresis. And then a third condition is synovial fluid treated with trypsin, which most of you may know, cleaves also molecules, explicitly glycoproteins. And what we wanted to do by treating synovial fluid with these different enzymes is a very simple model of pathophysiology, because in pathophysiology we have higher expression levels of different enzymes. So an enzyme that targets glycosaminoglycans, an enzyme that targets glycoproteins, period. And now what we see with the blue curve, loading and unloading, is that at certain regimes, the curves overlap, so little hysteresis or no hysteresis, but in other regimes, there's small hysteresis. So we repeat this over the same locations multiple times. Again, synovial fluid treated with trypsin, hyaluronidase, and long story short, because I want to touch based on the last study, we came up with very simple mechanical model that full, healthy synovial fluid, of course, under the investigated conditions, behaves as an elastic spring. If you remove hyaluronidase, we propose this behavior, including a dashboard, so you have a highly dissipative component there. You don't recover the properties of the film. And if you remove glycoproteins, the elastic performance or regime of that film is slightly reduced. Okay? And we published that a few years ago. Now, the part that is a little more interesting, and this is the first time I'm going to show you these results, show these results. It's on fibronectin. So now we're finding a little bit more the cartilage surface model. So there's no SFA involved here. So I'm going to introduce another technique. So in the 80s, Denninger publishes this micrograph showing that fibronectin is present. Fibronectin is another protein that is in cartilage, in addition to collagen type 2 and many other things. So what we're trying to do is to add fibronectin, which in this biotribology field, no one has really looked into. So we have collagens, we have aggregans, we have glycosaminoglycans. All of those have been integrated to different studies, but fibronectin has remained out of the picture. So if it's there, there's a reason for it. So that's the question that we're after. So very briefly, fibronectin, this is one dimer of fibronectin, it's a dimeric molecule, has domains that will drive or control the interaction with other molecules. That's the point. And depending on the conformation, if it's folded, some domains are going to be, of course, not accessible, cryptic, to molecules, and there's not going to be any interaction. But if the molecule changes the conformation, cryptic binding sites will be exposed, and there's going to be some interaction. So we want to start identifying what moieties of fibronectin, cryptic or not cryptic, are responsible for mediating synovial fluid film formation. And that film, at the end of the day, will have some consequences in the tribological performance. So for that, we used a quartz crystal microbalance. So this study is mostly quartz crystal microbalance. It's a crystal. We excite it at resonant frequencies. And this resonating crystal, we mount it in a fluidic device or cell, flow liquid, and then that liquid is loaded with a molecule of interest. And if the molecule absorbs, it is a simple harmonic oscillator. The frequency will shift. We monitor the shift in the frequency, and then we can correlate that shift in frequency to aerial mass. Very simple. Actually, very nice system. And this is our experimental setup. So we have fibronectin in solution, this large glycoprotein, and at neutral pH, it has a compact conformation, relatively inert, doesn't interact with many things. But we can use urea, or we can change the conformation also using pH. And acidic pHs, the conformation of fluorocarbon changes and becomes unfolded, okay? So what we're doing here is changing the conformation of fluorocarbon in solution, exposing it to our substrates, quartz crystals, with self-assembled monolayers, and immobilize the molecules, and then expose these protein or fluorocarbon films to full synovial fluid. And the idea here is to quantify the amount of fluorocarbon adsorbed to the surfaces, and then the amount of synovial fluid adsorbed to these fluorocarbon films, ideally with different conformations. We don't know if we're preserving the conformation. So we work with short self-assembled monolayers, and we work with long self-assembled monolayers, thinking that the gold substrate being too close to the protein can induce or have influence on the conformation, and we would lose control on the conformation imposed in bulk. By having a longer self-assembled monolayer, we are decoupling the immobilized protein from the surface influence. For example, van der Waals forces. So the first summary. Fibronectin, so we have on the left-hand side, fibronectin immobilized on short self-assembled monolayers and on long self-assembled monolayers. And I'm going to focus only on the pH 4, and I'm not going to go to this level of granularity. But we have fibronectin deposited on short at pH 4, so extended. So clearly we have more fibronectin on the film compared to fibronectin deposited from pH 7. So there's difference. So the conformation, or the amount at least, is different. We do the same on long self-assembled monolayers, and we see that denaturated or unfolded fibronectin amount is very similar to the short case, but the amount of fibronectin immobilized to the surface on long self-assembled monolayers at pH 7 is slightly higher than that than on short self-assembled monolayers. So in these conditions, we believe the substrate has an influence on the amount of fibronectin, potentially on the conformation, leading to the amount of self-fibronectin interactions. Very briefly, AFM, all reviewers love AFM images. We have some AFM images. So we have, again, short, pH 4, pH 7, long pH 4, long pH 7. And without going into much detail, we clearly see differences in the morphology. We have different topographies. And we believe that these domains below, and we have some, of course, controls, are features due to the gold topography. And these gold surfaces, I have to tell you, are extremely rough, around 3 nanometers. So there's ways to go around that and get rid of the topography by mica templating gold and having relatively smooth surfaces. But that's not something that we did here. So in collaboration with Kevin DeFrance at Queen's University, not far from here, they have this very special setup. So circular dichroism, working with polarized light, is a very common technique to quantify alpha, helix, beta sheet content, or disorder domains of proteins in solution. But they have the capability of also quantifying these tertiary and tertiary and quaternary structures of immobilized protein films. So we send them our quartz crystals with the different protein films, and they run these CV measurements. And what they observed are these different spectra. So we have, for short, we have the far UV and the near UV, and then we have the long sun, far UV, and near UV. And we have quantified this. Summary is, you can clearly see differences in the peaks, which correspond to random coils, beta sheet, or alpha sheet. The amounts are not only different between the pH values, but they're also different depending on the length of the sub-assembled monolayer that we use to immobilize them. It is more evident at this near UV, where quaternary structures can be easily identified, particularly phenylalanine, tyrosine, and tryptophan resonant peaks. And we see that they're shifted, for example, this peak to this peak. And because these conditions, some short, long here. This pH4, this pH7, this pH4, this pH7 in bulk were the same. These differences are clear due to the induced by the artifacts of the surface. So that's what we are interpreting here. Last but not least, we finally expose these protein films to synovial fluid to get an idea of how much. We are not interested, well, we are interested, but we don't care yet about what components of synovial fluid are absorbing to these protein films. And we are doing also some mechanical modeling to obtain the shear dependent compliance of these films, but that's possible with QCMD. But for simplicity, I'm focusing only on the Sauerbrei mass, or the masses. And we see that between short and long self-assembled monolayers, and also between the pH, there are differences in the amount of synovial fluid deposited. So with that, we came with this very simple proposed summary or mechanism. So we have on short self-assembled monolayers, short, we have a lot of fibronectin and a lot of synovial fluid adsorbed. And on short at pH 7, we have less fibronectin adsorbed. Of course, the conformation is more compact. And we have still a good amount of synovial fluid. So not a dramatic difference between these two conditions in terms of synovial fluid. And on long self-assembled monolayers, much longer, we have a lot of fibronectin in the pH 4 case, a little of fibronectin in the pH 7 case, and we have less synovial fluid adsorbed compared to the short self-assembled monolayer case. If this is physiologically relevant, I don't know. For sure, no. But what we can tell from this study is that it is important to look into the conformation, or more importantly, the distribution of populations of the molecular conformations of fibronectin at the surface of articular cartilage. Can we do that in vivo? We need to find ways to do that in vivo, okay? And I have some ideas, but that's coming next. And then investigate which components are actually absorbing to these precursor films. Of course, synergies. I spoke in the first part about synergies. We need to incorporate to these models collagen type 2, hyaluronic acid, agglicans, decorins, and some other components to really understand how a more complex extracellular matrix regulates the absorption of synovial fluid components, the formation of a lubricating, wear-protecting, and load-bearing film. So with that, this is clearly the wrong summary. So with that, I want to thank, of course, the group, funding agencies, and all of you for the attention at Friday evening, afternoon, late, anyway. So thank you for sticking. I have, of course, I'm open to receive any questions. Thank you, Alex.
All right, thank you. Let's thank you.